DNA to mRNA or DNA to what else? RNA, RNA, tRNA, etc. Ribosomal RNA as well. Good. And then what's translation in? All right, this is easy for you guys. Super. So let's start there. So let's just break down what we're going to talk about. DNA replication is just the replication, taking a double-stranded piece of DNA, making it single-stranded, splitting it apart, unzipping it, so to speak, and then laying down a whole new strand based on that template. Transcription is the conversion of DNA into RNA. Either a tRNA, a transfer RNA that moves those amino acids up to build the protein chain, or ribosomal RNA, which is an integral component of the ribosome, splitting ribosomal proteins, or the messenger RNA, which he said, which is right. We all think about the messenger RNA because that's the next step going up to protein synthesis and translation. So translation, though, uses all of those pieces. So translation takes that messenger RNA together with ribosomes and the loaded up tRNAs, the tRNAs that are loaded up with those amino acids, and builds a protein chain. Takes those amino acids and converts them, builds them into a long polymer, long polypeptide chain to turn it into an actual protein. So let's go through the steps of these. And hopefully this is, as I said, hopefully it's a pretty good review for everybody. If I do go too fast and I miss somebody, if it's been a while since some of you had your fundamental biology, then by all means raise your hand and I will happily repeat. It's very important that you get all of this easy stuff at the beginning. All right, so DNA is basically just a sugar backbone. So DNA is composed of either a ribose or a deoxyribose. RNA is composed of a ribose. DNA is composed of a deoxyribose, these sugars. They come together with the purine or the pyrimidine bases. What's purine and what's a pyrimidine? Or just, you don't even have to remember which is which. AGCT, you're right. Adenine, guanosine, cytosine, thymine, and tyrosine. All right, okay? And when they come together, when you get the sugar together with the purine, the pyrimidine, the base, that's what we talked about. You heard, remember base pairs. Base pairing is tying up those purine and pyrimidine bases. That's a nucleoside. Nucleoside in the case of RNA, or deoxynucleoside in the case of DNA. And once it's phosphorylated, it becomes a nucleotide. And DNA or RNA are just long chains of these nucleotides. All right, so in this case you have adenosine on a ribose sugar. That's RNA for ribose sugar. And there's the adenine, the purine base. All right. And if you phosphorylate it, in this case you have deoxyadenosine, where the 2' are missing that other group. Once you phosphorylate that, it becomes deoxyadenosine monophosphate. If you write that out, it would be AMP, but it's now a nucleotide instead of just a nucleoside. It's just jargon, it's just terminology. Jargon's not word. All right, so as most of you know, DNA it's a double-stranded and complementary strands, and it's complementary strands that are bound by those base pairs. And so you have anti-parallel base pairing, where the G's bind to, this is in DNA, the G's bind to the C's, and the A's bind to the T's. And you can cheat and look at the slide if you want, but what's distinctive about the fact that G binds to C and A binds to T? How would you know? What's important about that? Hydrogen bonds, excellent, it's done by hydrogen bonds. And how many hydrogen bonds are in a GC? 3, good, you can cheat and look at the slide. And how many hydrogen bonds are with when AT binds to each other?
2.
2, good. And why do we care about that? Oh, this is easy, there's no crack. Why?
It's easier to break the G.
Easy to break? What's another word for breaking? What do we do with DNA? Denature it. This. We melt it. Okay? Denaturing this, breaking it, all good words for melting it. Okay? And so DNA that has a whole lot of guanosine GC bonds has a whole lot of triple bonds holding it together, as opposed to DNA with AT bonds. Okay, and this is gonna come up a little later in the semester with respect to certain types of organisms. So organisms that have a very GC-rich DNA, and those of you that have spent a lot of time thinking about molecular biology, you're probably queuing in on what I'm saying. Organisms that have a lot of GC-rich DNA, their DNA is more stable at a higher temperature. So if I were doing a PCR reaction on the template strand of DNA or the primers that had a lot of GCs, I have to melt it at a higher temperature. And if you're not familiar with PCR and that didn't ring in your ears, don't worry, we'll have plenty of time this semester to get through that. Okay, let's talk about RNA briefly. Again, it's a polymer of nucleotides. Sometimes single-stranded, sometimes double-stranded. And let's just remind you, really simple, good basic review. You have a messenger RNA. It's the actual template that codes for the protein chain. You have ribosomal RNA. It's a component of the ribosomes. Ribosomes are a component of RNA itself and protein. And then you have transfer RNA, those big funky cross-like pieces of RNA that travel and take an amino acid directly down to the ribosome so it can turn into that, they're carriers, turn into that protein chain. In protein structure, if you're not remembering, it's just a big polymer of amino acids. Before we're done today, hopefully if we have time, we'll go through that, the structure of that and how it's formed. For this course, I do not expect you to memorize all the structures of all the amino acids. There's a ways to hope. Take biochemistry, take something else, and you'll have to memorize it. I'm just curious. Have you guys had a class yet that made you memorize all those things yet?
Yes.
I won't do it. You should have to memorize them. Let's write a passage. Oh, and speaking of that, something else I wanted to ask. Have you guys also had classes that made you memorize the Krebs cycle? I didn't make you memorize that.
Yes.
You've had to memorize the Krebs cycle before? Yeah, you should have. Okay, so let's jump into DNA replication. So as I said, DNA replication is just the unzipping of that double strand, the melting of it in a sense. Like in this case, it's melted enzymatically. To where you can lay down, you have a parental strand and a new strand. All right? And what happens is you unzip it, you have all those base pairs just hanging out there free. And then if you have free nucleotides in the environment, they'll in theory attach to it, right? And recreate another double strand. It's the natural state for this. And this always happens, as they unzip, new DNA is enzymatically laid down. It reads it in a 3 to 5 prime direction. So who remembers 3 and 5 prime, and what does that mean? Good, and they bond they're going in reverse direction. So it goes 3 to 5, 3 to 5, and it's connecting up in that way. I'm not saying this clearly. As these nucleotides build this long chain, they're connecting up 3 to a 5, 3 to a 5, 3 to a 5. This is the 3 prime, and up here is the 5 prime carbon. Okay? All right? And so this chain, in a sense, starts down in a 3 prime end, and it goes to a 5 prime, and 3 prime, and 5 prime, and 3 prime. So you always have trailing 3 prime end, and that's anti-parallel on the opposite side, where you have the 3 prime end down there and the 5 prime down there. All right, and that just refers to the number of the carbon in that sugar, in that deoxyribose sugar. Okay. All right, so this is important though. New DNA is laid down in red in the direction the enzymes move up the 3 to 5 prime direction. So on one strand, you have no problems, right? As it unzips down here at this 3 prime end, we're looking at this blue parental strand. As it unzips, it just reads right up and lays down new in an opposite direction. So that down here in the purple, you have the 5 prime hanging down. Okay? Well, what about on the other strand? Keep track of these blue. Here you have the 3 prime on the blue. There you have the blue with the parental. You have the 5 prime on the blue. But it's reading 3 to 5. So it can't start down here on the 5. It has to start up here at the 3 and go down. All right, and who remembers how that's handled? Anybody remember that?
Ogasaki fragment.
Okay, cool. A couple of you do. Not everybody does. That's okay. It's okay. A couple of you do. Okazaki fragments. Right. And And we'll talk about that in just a second, okay? So all these principles hold. We're showing you here in just a nice linear piece of DNA. It's really easy to think about. But remember that bacterial and archaeal chromosomes are in a circle. Still double-stranded. It's a double-stranded ring. And they're in a circle. And you have to basically unzip that circle. Now, we saw pictures of this in earlier lectures. So let's look at it again. Where you basically have this nice double-stranded piece. You have an origin of replication where that starts. And then you have the fork. The fork refers to the point at which those parental strands are unzipping from each other. And then the new strand is laid down again in the 3 to 5 prime direction, reading in the 3 to 5 prime. Oh, the new strand's coming at 5 to 3. And so you basically just unzip that ring and end up with 2 rings that eventually break apart. So the principle of that linear piece still holds. You just have to do it around a circle, and then you end up with 2 rings. Okay, so let's think about it. There's a lot of really nice enzymatic chemistry that goes on there to make replication take place. And there's some important proteins that help with this. And in just a slide, I'm going to give you one. We're going to talk about those Okazaki fragments for people that don't remember. And 2, we're going to kind of see how this mechanism, how the steps of this work. And so you have some important proteins. You have helicases, single-stranded binding proteins that pull them apart. Topoisomerases keep it from twisting, and I'll show you that in just a second. Likewise, DNA gyrases at topoisomerases. And primases are the ones that get the primer down there. When you need a primer, you need a little piece of known DNA to stick down there and extend and start to extend that new second strand, that new daughter strand. All right. So here you have your blue double-stranded parental strand that we're going to replicate. All right. And up top, the leading strand, you see it has its 3' end right there. And the lagging strand has its 5' end right there. Follow that blue line. All right. Some of the functions of these enzymes and proteins is that you have single-stranded binding proteins that pull these pieces apart and will actually bind down onto that template strand. DNA helicase is what unzips it with the replication fork. All right, and let's talk about DNA gyrase. So if I have this binding helix of double-stranded DNA, and as I unzip it, it's going to get tangled. So as I pull this apart, if I have something twisting, I always use my keys. Here's your double-stranded DNA. It's bound together. As I pull that apart and create a replication fork, this gets more and more tingled and twisted and tight. All right? As you're trying to unzip it, it's getting knotted up above the fork. And where it gets knotted up above the fork, you have an enzyme there that relieves that pressure. As I try to unzip something that's already twisted, it's going to get tangled up just above the fork. DNA gyrase relieves the pressure there and maintains no tension so that below the fork, the replication can take place. All right. So you have DNA primase that's getting a primer down there, a little RNA primer that starts the process of DNA replication. And DNA polymerase. Where's DNA polymerase? DNA polymerase is what actually extends the fragment. All right, but let's look at the differences in the strands. So again, you'll notice that on the leading strand, it's really easy. It starts at 3', and it lays down, it just reads as that fork open, it lays down more and more and more because it's reading from the 3 to 5' direction. 3', 5'. So it has no problems. On the lagging strand, though, it has to do it in little segments as this opens up. Okay? So it has to actually start its replication up here. Notice the primer is way upstream so that the new piece gets laid down in little fragments. And those little fragments are called the Okazaki fragments. All right? So as this opens up, this is just a sequence of events. As this opens up, you see the top leading strand easily replicated, and then you see little sections being replicated. And then in a minute, we're gonna see another section coming out right there. All right? But as this opens up, the 3' end is up here, 5' end is over here. These little sections just build little fragments, Okazaki fragments. And eventually they get found together. Yeah, Mike?
The SSBs mark the fragments, so will it go between them? 'Cause that's what that picture looks like.
That's what that picture looks like, and I don't remember for sure. I apologize for not having a better answer for that. I don't remember for sure. I know they're what pulls it apart.
Yeah.
But obviously you have, that's a really good question. Obviously you have, according to this cartoon, an SSB for each fragment. I don't remember the why. Okay, I'm sorry, I don't remember that. Alright, but eventually those Okazaki fragments have to get bound together so that you still end up with another second strand. And it eventually becomes continuous. So, easy, the leading strand, no problem, it just replicates in the flag. The lagging strand has to do with segment.
Question, DNA gyrates and topoisomerase, are they the same?
Yes, thank you for asking that. Sorry, I wasn't clear. DNA gyrase is a topoisomerase. We do the same thing. So helicase opens it and unzips it. Gyrase keeps it from getting tangled up into a knot. The single-stranded binding proteins keep those strands apart. The trimase lays down the primer. Alright. We just don't show a picture of the enzyme. And DNA polymerase extends out that link, just like those of you that have done a PCR reaction. Okay, more questions? Okay, so let's kind of go through these 3 steps then. Let's jump into transcription and translation. So, as we like to say, the point of transcription is to convert your DNA into RNA. But you can't do much with your DNA unless it becomes RNA at some point. Whether it's ribosomal RNA, tRNA, or a messenger. In this case, because we're creating a protein chain, it's going to be a messenger in this cartoon. Okay. So you have a coding strand and a template strand. And not that different than when you replicate DNA, it gets read from the 3' to 5' direction. It starts the process of transcription at the 3' end of your template strand of DNA. And it lays down messenger RNA from the 5 to 3'. Alright, so in DNA, when you have base pairs coming together, you have A's coming to T's, C's coming to G's, etc. In RNA, you don't have thymidine, you have uracil. So you have AU coming together. So here you have the template strand, and you're laying down a new messenger. So it's TA, GC, AU, CG, etc. It's an important little fact. And I'll show you, we're going to go into more detail on this in just a minute. The template strand lays down that gene has distinctive patterns in these little codons. Okay? AUG always codes for a start codon. Anyone remember what amino acid that is? AUG? Fine, good. It's going to be a little different for bacteria, but yes, fine, good. And then each 3 nucleotides are codons for particular amino acids. Alright, so this is transcription, turning it into RNA, and then we just know that each 3' threesome are codons for a particular amino acid. And those codons go through translation to have anticodons on the tRNA. So I'll explain to you in just a minute why each one doesn't have its own, but an amino acid is going to have its own codon on the tRNA. Each one of them has exactly the same. It binds an basically opposite of these codons. So methionine is the first one, AUG, like we said, methionine, and then its anticodon on the tRNA is UAC, etc. And I'll show you how these things bind up to build a long amino acid chain. We'll go through that before the end of the lecture. Okay, so there's distinctive features to a gene in its DNA, and then logically into its RNA. All right, so again, here we have the coding strand and the template strand. You have a promoter region, all right, and that's where the RNA polymerase will find the gene to create the RNA. And notice the numbering scheme. So transcription actually starts, the RNA polymerase finds it here, but transcription actually starts at the 0 marker for that gene, okay? And we number that, it's very simple, we number it based on nucleotides, okay? So here we have the start codon, then that would be 1, 2, 3, 4, 5, 6, 7 nucleotides in, all right? And then upstream of that are negatives. So for instance, the point at which RNA polymerase binds is at -10, recognizes at -35 on this figure. All right, and then you have a leader, and then you have the coding region, which the start codon is actually right there. All right, so then it numbers out from there. All right, but again, just like in DNA replication, in transcription, it reads 3' to 5' to lay down an antiparallel mRNA, in this case, that's 5' to 3'. Okay. So, RNA polymerase binding site, leader, start codon, your mRNA, your protein translation is going to start right there. Okay, so let's go into kind of a little bit more details on transcription. Okay, so in the same way, transcription has to unzip that double-stranded DNA. There's two important things here. Here's RNA polymerase, which is going to generate the new RNA strand. That's the big enzyme that's going to wrap around that DNA, that single strand of DNA that's been unzipped, and going to generate a new messenger RNA or RNA strand, tRNA. And then we have something that we're going to talk about more later in the semester, but we need to talk about it today too, which is a sigma factor. Anyone here heard of a sigma factor? Interesting. Those of you that have genetics, they don't talk about sigma factors in genetics. Maybe I'll just quiet. What a sigma factor does is a sigma factor guides your RNA polymerase to the correct spot on the gene. And sigma factors, we're going to spend more time on this later, but they're global regulators. So for instance, if I'm a bacterial cell and I'm going into stationary phase, there is a specific sigma factor, sigma S in this case, don't worry about that, but there is a specific sigma factor that can be coded for that will basically guide RNA polymerase to all the necessary genes and will upregulate the genes necessary for a cell to survive stationary phase. Likewise, there's, say a bacillus cell wants to go into sporulation. They're starved, they're hungry. You guys have dealt with this in lab. They want to go into sporulation. There's also specific sigma factors that will guide the RNA polymerase to the genes so that all the right proteins get expressed and ultimately translated to form sporulation spores in the cell. So the sigma factor guides RNA polymerase there to that template strand, again in the 3 to 5 direction. It unwinds it, and it lays down new messenger. Okay, so this new messenger is coming off, the 5' end is coming off first because it read it from the 3'. Once transcription is initiated, the sigma factor can leave. The sigma factor's job is just to carry the RNA polymerase to the right spot. And once that happens, it can leave. And then the RNA polymerase moves down that DNA strand and it rewinds on the flip side, leaving out this big long messenger RNA transcript. Okay, so let's look at that enzyme up close. Okay, so again, you have the RNA polymerase binding sites. This factor, here's the whole RNA polymerase enzyme. It basically forms a complex around that DNA. It opens up that DNA and then reads in the 3 to 5 prime direction, laying out this red tape as the messenger that's coming off. At this point, once transcription starts, the sigma factor can leave, and you end up with an RNA transcript coming off. You can see it up close. You can see that it's like a tape. The messenger RNA coming out at the 5' end. Ribosomes can automatically attach to that even before transcription is finished here in this, called a transcription bubble. Ribosomes can already attach to this and start protein synthesis as that messenger is being formed. We'll talk about that in just a second. Okay. So let's go up to translation. The translation is the conversion of that messenger RNA code into a protein chain. And it does this by, let's see, shut the door. Let's do some talking. Thank you, I appreciate it. Okay. All right, the genetic code. So remember I said that the codons come in 3 nucleotide chunks. And each of those 3 nucleotide chunks codes for a particular amino acid. All right, AUG is always the start codon. And the way to read this chart is that you have the first position, the second position, and the third position. All right? And so you have, here you have everything starting with U, starting with C, starting with A, starting with G. The second position could be U, C, A, or G, etc. And what you'll notice is a couple of things. So each one of these threesomes codes for an amino acid. But one of the first things you notice is you have 64 different codons, but only really 20 amino acids. And so there's redundancy in the code. Code is degenerate in a sense that you can have, if you have CU of any kind with any third nucleotide base at the third spot, you're still going to get leucine. All right, valine, GU, but any U, C, A in the third spot doesn't matter. You're still going to get a valine. All right, so that's what we mean by code degeneracy in the sense that once you're in the third spot of a codon, it rarely matters what that nucleotide is. This is going to be really important when we think about mutations. Some mutations are completely silent because even if you change a nucleotide in that third spot, it doesn't matter. You're still going to end up with a valine in that case. All right. And it also matters to something we call wobble, which is that you can have tRNAs can be flexible. So think about this from a cell's perspective. If a cell has to make all these different tRNAs, so in a sense we have a code here with 64 possible different codons. So you would think you need 64 possible different tRNA anticodons, but you don't, because valine, leucine, proline, threonine, etc., many of them are extremely flexible about that third position. So what you'll see in your tRNAs instead is that it doesn't really care what's in the third position. And you'll have multiple different, you'll only have one different anticodon, CCG, to give you glycine, combined with either GGU, which doesn't make sense, G and U don't normally bind to each other, or GGC, etc. So they're very flexible in that, so that you don't end up needing 64 different tRNAs. You need far, far fewer, which is a much more economical thing for the cell. It just doesn't really care what's in the third position. So it very likely doesn't matter. So that's the codon that you have, those are the codes that you have on your RNA. How do we convert that into a protein chain? So here again, you have that transcription bubble in the RNA polymerase where you have DNA opened up and leading out a messenger RNA strand. So in this case, protein synthesis begins begins, in 3 5, yeah. It begins, yes, in the 5 to 3 prime direction. Okay, so you end up with what's called polyribosomal complex, polysome, where the RNA, the ribosome, excuse me, is in a long chain down that messenger RNA. And let me show you that up close. All right, so you have a ribosome moving through with the messenger RNA. You have elongation factors that are going to extend that out. You have initiation factors. I'm going to show you those. Remember, the bacterial ribosome is 70S.
Bless you.
So let's go through initiation of protein synthesis. You need a couple of things to come together for protein synthesis to start happening, and translation. You need the 30S subunit of the ribosome. Sorry, that's why we didn't need to go through so fast through that. Remember, the ribosome is 30S plus 50S, so it has a small subunit and a larger subunit. You need the 30S subunit to come together with that messenger RNA and initiation factors. So these figures are excellent when you're using them to study. They're really good, but go ahead and just make a little note in your notes. I don't expect you to memorize the function of every single initiation factor or elongation factor. I don't expect you guys to. All right, so the 30S subunit has to come together with the messenger and the initiation factors that help bring the, in this case, formal methionine tRNA. Remember, the start codon is methionine, AUT. In the case of bacterial translation, it's formal methionine. It's a little bit different, but it's still methionine. So all these things have to come together. You have to have that start codon loaded methionine tRNA. You have to have the messenger, and you have to have the 30S subunit. So they all come together. Once these pieces are together, the initiation factors can be released, and the 50S subunit can combine. So you have the full 70S initiation complex here, where you have the formal methionine and the P site. I'll explain these sites in just a second. And then you have your messenger RNA here, going through, 5' direction in the first, 3' direction tailing out. And then you have these 3 sites. You have the E site, the P site, and the A site. Anyone remember what those 3 sites do? They do? Anyone else? Okay, let's go through that. The P site is the donor site. This one that's not terribly intuitively clear. The A site is the acceptor site. A little bit more intuitive. The E site is the exit site. So every time a new tRNA loaded comes in to match with an anticodon with the 3 nucleotide codon on the messenger, it comes in at the P site in the middle. Alright? And it has its amino acid there. Alright? And let's go through elongation. So this is the same thing you just saw. I always put it up there because it's easy to refer back to it. Okay. So here you have a loaded tRNA in the P site. We call it the donor site because it's from the P site.